Hello, welcome to my YouTube channel. My name is Venon Software, and in today's video, I'm going to talk about the Pentose Phosphate Pathway. The Pentose Phosphate Pathway, it is known by many names. Sometimes it is called the Dickens or Recker pathway. It is also called the exose monophosphate shunt, also sometimes called phosphogluconate oxidative pathway. So this pathway, it occurs specifically in the cytosol or the cells. It doesn't occur in the mitochondria. It occurs in the cytoplasm, cytosol or the cells. So this pathway, it is most common in cells that produce lipids, where lipogenesis occurs, for example, in adipocytes. So this pathway, it also occurs in cells like hepatocytes as well as erythrocytes, which are red blood cells. So this pathway, it is very important for the production of NADPH. So NADPH is a very powerful reducing agent, or in other words, a very powerful electron carrier. So NADPH is produced in this pathway. What else are produced in this pathway? Ribose 5-phosphate. We can see it there. So ribose 5-phosphate is also very important. And what else is produced in this pathway? We've got glyceraldehyde 3-phosphate, which is there, and also fructose 6-phosphate. So NADPH is used in the synthesis of fat acids, as well as the production of cholesterol, the synthesis of neurotransmitters, as well as the production of steroids. So NADPH, it is also important in red blood cells to help detoxifying hydrogen peroxides, or in other words, detoxifying reactive oxygen species. Okay, what about ribose 5-phosphate? Ribose 5-phosphate is very important in cells and the production of nucleic acids. So this is a precursor for the production of nucleic acids. It is also important in the formation of coenzymes such as coenzyme A, and also in the production of NAD+ and also FAD. Okay, what about glyceraldehyde 3-phosphate and fructose 6-phosphate? So we can see that these are glycolytic intermediates. So what happens when these are produced? They immediately enter glycolysis for the production of ATP. Okay, now what happens if we don't need ATP? So if we don't need ATP, we are going to find that glycolysis will be inhibited, and that is going to result in glucose 6-phosphate to be shunted into pentose phosphate pathway. So if we don't need any more ATP, the board or the cell is going to be like, oh man, I don't need ATP. I've got enough ATP. So now, what is the board going to do with the excess glucose? It's going to convert the excess glucose to glucose 6-phosphate and then shunted into the pentose phosphate pathway for the production of NADPH, ribose 5-phosphate, and the glycolytic intermediates. So now, the whole purpose of this video is to talk about the medical correlations of this pathway. What is the medical significance of this pathway? So first and foremost, I'm going to talk about the use of NADPH. So I'm going to start with the physiological aspect before I go to the pathological aspect. So what is the law of NADPH? So here we have seen that we've got this enzyme, which is called G6PD. So G6PD simply stands for glucose 6-phosphate dehydrogenase. Whenever you see an enzyme which is having dehydrogenase in its name, just know that that enzyme, it is involved in the removal of hydrogen, or in other words, in the removal of an electron, or you can say in the removal of an hydride ion. What is an hydride ion? It is simply a proton with 2 electrons. So this G6PD is going to remove an hydride ion from glucose 6-phosphate and adds it to NADP+, so that it becomes NADPH. And NADPH is very important. Now imagine that this process, it is occurring in the red blood cells, erythrocytes. Okay, like I said, I'm not going to waste time to start talking about this pathway. I'm just going straight now to the medical significance of this pathway. All right, like I said, I'm talking about the physiological aspect now. So G6PD is important in the production of NADPH. What is the role of NADPH? So NADPH is important. For example, let's say red blood cells. We know that in red blood cells, each and every time, a lot of reactive oxygen species are being produced. For example, in hemoglobin autooxidation, we know that when hemoglobin, when it binds to oxygen and takes it to the tissue, when it releases oxygen, oxygen becomes superoxide. In other words, oxygen gains an electron. So oxygen, when it is released, sometimes it becomes superoxide in that state. So this superoxide can damage what? Can damage the hemoglobin. In other words, it can damage the cell, the red blood cell. So this superoxide, it must be neutralized by a molecule which is called glutathione. And also, when hemoglobin releases this molecule, which is the superoxide molecule is going to become methemoglobin. What do we mean when you say methemoglobin? So we can see here that normally in hemoglobin we have got iron in its feller state, Fe2+. So iron normally in hemoglobin, it is in its feller state. So when it binds to oxygen, takes oxygen to the tissue, it's going to release that oxygen in form of superoxide and it's going to become methemoglobin. So methemoglobin has got the ferric state of iron, Fe3+, because iron has lost an electron. So it's going to have 3 positive charge. Okay, so for hemoglobin to work, it can't work in this state. It needs to be converted back to this state, to the hemoglobin state, from methemoglobin to hemoglobin state. So here, for it to be converted to this, which means that this ion must gain an electron, so it becomes Fe2 positive. Where is that electron going to come from? It's going to come from NADPH. So NADPH, because like I said, NADPH is a reducing agent. In other words, it's an electron carrier. So it has got an extra electron in its hydride ion, which is this one. So NADPH is going to donate an electron to this ion, so that ion now can become in its ferrous state like that. And that is hemoglobin. So this cycle continues. So as this hemoglobin is working, it continues producing superoxides. So these superoxides, they can damage the red blood cells. So they need to be neutralized by another mechanism. So this mechanism also requires NADPH. So we can see that this NADPH is important. How? It is important in converting methemoglobin back to hemoglobin. And it is also important in neutralizing the superoxides or reactive oxygen species that are being produced in this hemoglobin autooxidation. All right, just a recap. Glucose 6-phosphate is converted to 6-phosphogluconate via an enzyme G6PD. In this process, NADP is converted to NADPH. What does NADPH do? NADPH is an electron carrier, or in other words, it's a reducing agent. So what is it going to do? It is going to transfer or to release its electron and donate it to what? To which molecule? To glutathione. So glutathione normally it exists in its oxidized state. So it has got sulfur bonds there. So disulfide bond there. We can see this is sulfur and this is sulfur. G is representing glutathione. So in between it has got sulfur. So this sulfur they form disulfide bond. So in that state we call it that it is in its oxidized state. So for this to work, for this glutathione to work, it needs to be in its reduced state. GSH. So it needs to have that thiol group. SH is a thiol group. So now, how is it going to be converted from its oxidized state to its reduced state? That's where NADPH is needed. So this guy, he'll be like, oh, I need an electron. I need an hydrogen to become like that. So you can see here it doesn't have a hydrogen. There it has got an hydrogen, GSH. So now, where is that hydrogen coming from? There'll be this enzyme, this good dude. It'll come, glutathione reductase, is going to get an hydrogen from NADPH, steal that hydrogen and adds it to what? To glutathione there, and it becomes in its reduced state like that. So glutathione reductase is going to get hydrogen ion, or in other words, an electron from NADPH and adds it to glutathione, so it becomes in its reduced state like that. So glutathione, when it is reduced, it has got an important function in red blood cells. What does it do? It is going to neutralize these toxins I was talking about, the superoxide, the hydrogen peroxide, and other reactive oxygen species. All right, how is it going to do that? So there's another dude here, glutathione peroxidase. So this guy is a very good guy. What does it do? This guy is going to transfer this hydrogen from the reduced glutathione, and it's going to add it, bring it to what? To this guy, hydrogen peroxide. So it's going to get this hydrogen and adds it to hydrogen peroxide. So hydrogen peroxide now, which is the bad guy, will be converted to water. So this guy now is going to be powerless when it's converted to water. It can damage the red blood cell or the cell generally. So this glutathione peroxidase will get this hydrogen, or in other words, will get an electron from the reduced glutathione and adds it to hydrogen peroxide. So it becomes H2O as well as oxygen molecule. So in that way, NADPH is serving a role in detoxifying these reactive oxygen species in the red blood cells. So physiologically, you're going to find that this red blood cell is going to work perfectly. Okay, what's the other use of NADPH? NADPH is also important. Where? In white blood cells. So for example, imagine this is the guy, the neutrophil, or imagine this is the macrophage. So in the macrophages, what happens that oxygen is being converted to its superoxide form, or hydrogen is being converted to superoxide. So via an enzyme which is called NADPH oxidase. So NADPH oxidase, what does it do? It converts oxygen to this superoxide. And this superoxide is used by the macrophages, for example, neutrophil. When they engulf a bacteria, they are going to start releasing this superoxide, reactive oxygen species. And those reactive oxygen species, they are going to kill this bacteria. Okay, and that way, they are going to emit this bad guy. So in other words, we can say that NADPH will donate an electron to oxygen, and then oxygen will be converted to what? To superoxide. Is going to have that electron which is coming from NADPH. And this superoxide can either be converted to hydrogen peroxide, or it can be used like that in what? In phagocytosis, and kill this bacteria by the neutrophils. So okay, just a summary. I've talked about the use of NADPH. NADPH is used in inducing glutathione, so that glutathione can convert these reactive oxygen species to water and oxygen. And then it is also used in converting methemoglobin back to hemoglobin, so that hemoglobin can transfer oxygen. Okay, and it is also used where? In white blood cells, in the process of phagocytosis, where these foreign substances or foreign particles are killed. All right, that's a very wonderful use of NADPH. So physiologically, it's important in that way. Now, what can happen if someone is not having G6PD? Like I said, G6PD, it is very important in the production of NADPH in the first step as well as in the third step of this process, which is pentose phosphate pathway. So G6PD, it is important in production of NADPH. So there, even here we have got 6-PGD, which is 6-phosphogluconate dehydrogenase. It produces NADPH. There, G6PD produces NADPH. Now, imagine what can happen if someone doesn't have G6PD. If someone doesn't have G6PD, this process won't occur. Everything won't occur this side, right? So someone who is deficient in glucose 6-phosphate dehydrogenase, that person won't be able to produce NADPH. And remember, we said that NADPH is used in fat acid synthesis, production of cholesterol, and also it helps in eliminating these reactive oxygen species. For example, in red blood cells. So what will happen if someone doesn't have G6PD? This person won't have what? NADPH. Okay, let us look at this diagram again. So someone doesn't have G6PD, this person won't be able to produce NADPH. And this person can't produce NADPH. This guy, glutathione reductase, it won't work because it doesn't have this guy, NADPH. It doesn't have a source of an electron. So if this guy won't work, then glutathione will remain in its oxidized state, won't be able to be reduced. What will happen? If glutathione won't be reduced, what's going to happen? The implication is that this guy here, this dude, glutathione peroxidase, is not going to work as well. And if this guy can't work, what does it mean? Is that this glutathione can't convert hydrogen peroxide, which is toxic to the cell, to water and oxygen. So this glutathione peroxidase can't convert reactive oxygen species to H2O and O2. Okay, this is very devastating to the cell. For example, to the red blood cell. Okay, so what's going to happen to the red blood cell? So now the implication that's going to happen is that this hydrogen peroxide, if it can't be converted to water, is going to start building up in the cell. And when it starts building up, it is going to cause what? Damage to the cell membrane. It's going to cause damage to what? Damage to the enzymes in the red blood cell. And we know that the red blood cells, they don't have mitochondria. They don't have ribosomes. These cells, they also don't have nucleus. So once the enzymes are being damaged, they can't be replaced because they don't have the ribosomes, they don't have the nucleus. So these enzymes that are going to be destroyed, they can't be replaced. And for example, imagine that, for example, the calcium channels, the sodium, potassium, ATPases, pumps, all those are damaged. It's going to cause an influx of more and more charged particles into the red blood cell, and the red blood cell will be completely damaged. And then, what is the other thing that's going to happen? In the red blood cells, we know that there is hemoglobin. Hemoglobin, when it binds to oxygen, is converted to methemoglobin. So for it to go back to hemoglobin, it needs NADPH, which is produced by G6PD. Now, in the absence of NADPH, hemoglobin won't be converted to what? Won't be converted back to this. Methemoglobin won't be converted to hemoglobin, in other words. So in the absence of NADPH, methemoglobin will remain in that state. It can't be converted to hemoglobin, and methemoglobin cannot bind to oxygen. So we'll find that hemoglobin will remain stuck in methemoglobin, and it can't work anymore. So people lacking NADPH, you're going to find that the hemoglobin will be in methemoglobin, and it's going to have another implication. What else is going to happen? I said that NADPH is important in what? In white blood cells. Specifically, I said neutrophils and macrophages. So what's going to happen? You're going to find that in the absence of NADPH, these neutrophils, they can't produce what? They can't produce the reactive oxygen species, which are required to eliminate pathogens. So it's going to have an implication as well on the immune system. All right. Now, just going back to this again, to the buildup of hydrogen peroxide in the cell. I said it's going to damage the enzymes. Once enzymes are damaged, you're going to find that this cell is going to die prematurely. This red blood cell is going to be taken into the spleen. So it'll be taken to the spleen, and there it's going to be killed. So what happens when taken to the spleen? Like I said, hemoglobin will be stuck in methemoglobin. When it is stuck in methemoglobin, what follows? You're going to find that this methemoglobin is going to bind to the plasma membrane, and it can't transport any more oxygen, because the only hemoglobin, or the only thing that transports oxygen is hemoglobin in that state, not in this state, not at all. So methemoglobin will bind to the plasma membrane and it can't work. So now these are what we call Heinz bodies. So when you take peripheral blood smear, you're going to see Heinz bodies under your microscope. So this is how they look. So these are Heinz bodies. So we can see hemoglobin, those dots there, bluish dots, that's hemoglobin, which has bound to the plasma membrane. And that hemoglobin can't work anymore. So those bluish dots, those are Heinz bodies. Okay, so that is hemoglobin is stuck in its methemoglobin state. It can't be converted to what? To its normal state, and therefore it can't take or transport oxygen. So in people who lack G6PD, you are going to see Heinz bodies. What else are you going to see? You're going to see bite cells like this. These are bite cells. So what happens? Macrophages, they come and chop or they bite that Heinz body. And when they bite that Heinz body, what's going to happen? The cell is going to remain like that. So they're going to bite Heinz body. And then when this cell is taken to the spleen, it's going to be digested or broken down into heme and globin. So the hemoglobin will be digested into heme and globin in the spleen. That's why this digestion occurs. Okay, like I said, the buildup of reactive oxygen species will cause premature death of this red blood cell, because we know that normal red blood cell, they take 120 days to die, approximately 120 days. So now, if this cell is having hydrogen peroxide, the macrophages will detect, like, oh, this cell is infected, this cell needs to be eliminated and be taken into the spleen and be digested. Okay, when digested, it will be broken down into what? Into bilirubin and conjugated bilirubin, as well as the production of what? Of heme and globin. So hemoglobin or methemoglobin, in other words, rather, it will be converted to heme and globin, and then there will be also production of unconjugated bilirubin. So now, what will happen to bilirubin? Bilirubin will be taken into the liver in hepatocytes, where it's going to be conjugated by the addition of glucuronic acid. Okay, I've talked about that, right? Now, the immune system is going to do another thing. You know what? The body is very good because it has got the process called homeostasis. So whenever there is destruction of red blood cells, it's going to start producing new cells. instantly. But now, those cells that are going to be produced, they are going to be baby cells, which is very bad for this patient, right? Okay, it's very bad indeed. Because baby cells, they can't transport enough oxygen. They are not yet mature. So these baby cells, they are called reticulocytes. So in people like G6PD, you're going to find out that in their peripheral blood smear, they have got reticulocytes. This is how they look. So you're going to see these kind of cells, these kind of cells with that purple mesh inside. Okay, these are reticulocytes. They are produced in the bone marrow. So these are baby cells. Before they grow, they are produced because the body wants to replace the dying cells, the dying adult cells. Okay, now we have talked about the downside of this part. So what I've just simply talked about, the premature death of red blood cells, if it continues, it can lead to what? It can lead to hemolytic anemia. So hemolytic anemia, it is caused because there is hemolysis. What is hemolysis? This is the destruction of red blood cells. So if red blood cells are being killed each and every time, it will lead to hemolytic anemia. So we are going to have a decreased number of mature red blood cells that can transport oxygen to the tissues. And this will result in other complications. So hemolytic anemia can be due to G6PD deficiency in the mechanism that I've just explained here. So through this mechanism, it can lead to hemolytic anemia. Now, this deficiency of glucose 6-phosphate dehydrogenase, it is also important in some way. It is also useful. How? Because like I said, lack of NADPH will mean that glutathione can't be reduced. If this can't be reduced, what's going to happen? You're going to find that hydrogen peroxide can't be detoxified to H2O and oxygen. So in the red blood cell, we are going to find that there is a build-up of hydrogen peroxide. Like I said, if this build-up of hydrogen peroxide, if it causes denaturalization or denaturation of enzymes, it is going to cause hemolytic anemia finally, right? But now, the good side of it is that these people who have got G6PD deficiency, they are resistant to malaria. Because remember, the parasite for malaria, Plasmodium falciparum, it lives and matures in the red blood cells. So now imagine that this parasite enters the red blood cells and it finds that in this red blood cell there are toxins. There is an abundance of toxins. There is a lot of hydrogen peroxide. What's going to happen? It's going to find this environment very bad for it. It can't even survive. It's going to die right there. So the Plasmodium falciparum, when it enters red blood cells, it finds that there's a lot of hydrogen peroxide, a lot of reactive oxygen species. It's going to die. It can't survive. Because the thing that you should understand is that Plasmodium falciparum, the parasite for malaria, it lives and matures in red blood cells. When it grows up in red blood cells, it starts killing red blood cells, breaking them down. That's why you see that patients who have got malaria, they lack blood, because this Plasmodium falciparum, it starts the lysis of red blood cells. So now, when it enters red blood cell to find hydrogen peroxide is there, and that's very bad for it. It's gonna die right there, which is very good. Okay, so these people who lack G6PD, they are also resistant to malaria. Oh wow, that's very good. But the downside of it, like I said, that these people like G6PD, they develop hemolytic anemia. But what you should understand is that this hemolytic anemia, it is asymptomatic. It doesn't show at all. In many patients, it doesn't show at all. Until you take some triggers, some oxidative stressors or oxidative agents. So if you take some oxidative stressors, for example, fava beans, which is also known as broad beans. So when you take these beans, what's going to happen? It's going to increase the oxidative stress. In other words, it's going to increase the production of hydrogen peroxide in the red blood cell. And that increase in the hydrogen peroxide in the red blood cell will trigger the lysis of these red blood cells. So these red blood cells will start now be quickly or prematurely being killed by the macrophages in the spleen. And that's going to trigger hemolytic anemia. So someone takes fava beans. It's going to trigger the production of more and more and more hydrogen peroxide in the red blood cell, which will lead to an increase in the lysis of red blood cells, and finally leads to hemolytic anemia. What else? Taking of some drugs, for example, primaquine. Primaquine is a drug that was developed 70 years ago, and this drug, what it actually does is that it increases the oxidative stress in the red blood cell, because this drug is an anti-malarial drug. It's a drug for malaria. So the way this drug works is that it increases the production of reactive oxygen species in the red blood cell. Because like I said earlier, I said that red blood cells, they are the one that contains the Plasmodium falciparum, the parasite for malaria. That's where it lives. So this drug, primaquine, it targets the red blood cells. When it targets the red blood cells, it increases the production of hydrogen peroxide, so that hydrogen peroxide can finally kill this Plasmodium falciparum. So this drug, primaquine, which is the drug for malaria, it actually increases the production of reactive oxygen species so that they can destroy this parasite, the Plasmodium falciparum. So now what's going to happen? What's going to happen in these people who doesn't have G6PD? When this drug, primaquine, it increases the production of H2O2, hydrogen peroxide, you are going to find that this hydrogen peroxide can't be neutralized afterward because this mechanism is lacking due to lack of G6PD. So you find that there will be an accumulation of hydrogen peroxide, more production of it, and this will trigger hemolysis or hemolytic anemia. Because this hydrogen peroxide can't be reduced. In other words, it can't be converted to what? To water. There is a lack of this mechanism. In normal people, if you take this drug, primaquine, it doesn't cause any problem. Because immediately this Plasmodium falciparum is damaged or it is eliminated, the body itself is going to use this mechanism and neutralize the produced hydrogen peroxide or the produced reactive oxygen species to H2O. But people like this, they can't neutralize the produced reactive oxygen species. So you find that primaquine will increase the oxidative stress and finally it will lead to hemolytic anemia. What else? Other drugs, so other sulfur drugs, for example, sulfamethoxazole. This drug, it also increases oxidative stress, the production of more reactive oxygen species, which can trigger hemolytic anemia. And also some infections. So infections, for example, pneumonia infection, hepatitis A, those infections can also lead to an increase in oxidative stress. So we've actually talked about hemolytic anemia and malaria. But now, how can we manage patients who have got hemolytic anemia due to G6PD deficiency? So the management actually is just to avoid the triggers, the oxidative agents. That's the best management. So we need to avoid, for example, fava beans, which can trigger hemolysis. We need to avoid things like sulfamethoxazole, a drug which can cause oxidative stress in red blood cells and eventually lead to hemolysis. We need to avoid drugs like primaquine. So the best way to do it, we need to screen a patient before giving them these drugs. Before prescribing them to them these drugs, we need to make sure that we screen them and see if they have a deficiency in G6PD. If they have a deficiency in G6PD, don't dare to give them these drugs that I've mentioned because it can cause hemolytic anemia. And also in some cases, this condition, or in other words, G6PD deficiency, it can be managed by blood transfusion. So for example, especially in babies, neonates, what we can do is that we can transfuse our blood into them so that now they have got new cells that have got G6PD. They have red blood cells that have got G6PD. But what you should understand that G6PD deficiency, it is an X-linked recessive inherited disorder. So this G6PD, it is more common in males than females because we know that males only have one X and one Y. So this is an X-linked genetic disorder. So when a male patient or a male has got this chromosome which is damaged or which is lacking the gene for G6PD, immediately that person is going to develop G6PD deficiency. Okay, but for females, it requires for them to be homozygous for them to develop this. They need to have this deficiency on both genes, both X and X, because you know that females, they have got two X chromosomes. Male has got only one X chromosome. So it is more common in male than females. So I think, guys, this is all I got for you. And please don't forget to subscribe to my YouTube channel for more good videos. Thank you for your support and for watching this video. Thank you so much.